Guys, let's complete section 8.3 DNA replication. After knowing the meaning of the DNA replication in the previous video, we know that the DNA replication is the copying of the DNA and it takes place during S phase. Let's know the steps of DNA replication. DNA replication starts at the origin of replication, a certain point at which the enzymes start to prepare the DNA for replication. How should this DNA be prepared for replication? Can it be replicated in the form of double helix, two strands that are connected together by the hydrogen bonding? Of course not. So at the origin of replication, enzymes will start to unzip the DNA double helix by breaking down the hydrogen bonding between bases. Here, look, now the two strands, the two original strands, the blue ones, they are separated by breaking down this hydrogen bonding by the help of enzymes. Another group of enzymes called polymerases or polymerase enzymes. Polymerase enzymes will start to form the new strand by using the original one or the old one as the template. As I told you before, in DNA replication, the cell will use one template, one strand as a template for the formation of the new one. This orange one is the new. The orange one is the new. What the role of DNA polymerases or DNA polymerase enzyme to add nucleotides? From where are those nucleotides? Those nucleotides are free floating in the cell. Those nucleotides are free floating in the nucleus. So by adding the nucleotides and by binding the two strands together by hydrogen bonding through the bond between bases, and also the DNA polymerases form covalent bonding to form a new strand. If you remember, covalent bonds bind or connect the nucleotides together in the same strand, while hydrogen bonding between two strands to form the double helix. So that DNA polymerase enzymes bond the nucleotides together to form the double helix, and polymerase enzymes also, they form covalent bonds between nucleotides. Now, there are two strands, two new double helix. Each one contains one original strand and one new strand. One original, the blue one, and the orange one is the new one. That's why DNA replication is a semiconservative process. So my question now, why is DNA replication semiconservative process? Because after DNA replication, two new molecules of DNA are formed, each with an original strand and a newly formed strand. That's why it's a semiconservative process. After that, the process of replication actually is so fast and accurate. Why is it fast? It's fast because it starts at many points in eukaryotic chromosomes. There are many points or many origins of replications. That's why this process is fast. Try to imagine if we have a project. What if I told only one girl to do all the project? Will she be fast? Or if I told many girls to take different tasks? Of course, if I asked many girls to start at a certain point to take different tasks, in this case, the process or the project will finish faster. Finally, the replication process is an accurate process because DNA polymerases, after adding nucleotides and after making hydrogen bondings and covalent bondings, the DNA polymerases also, they check errors and correct errors. That's why it's an accurate process. So now we know that replication process is fast and accurate, and we know the causes. Now we finished the process of replication. Thank you.